Okay, so today we are going to be taking a look at how the product or the result of glycolysis, which is the 3-pyruvate molecules, is turned into the precursor molecule for the all-important metabolic pathway, the Krebs cycle or the citric acid cycle, which is, as we know, the citric acid cycle is ultimately going to give us this our high energy ATP molecules. So we're going to talk about the reactions and the five cofactors that turn this 1,2,3 carbon pyruvate into this 1,2 carbon acetyl CoA or acetyl coenzyme, with the coenzyme basically being able to transport the acetyl group to start the Krebs cycle. So in order for step one to occur, the pyruvate molecules are going to be hanging out in the cytosol of the cell, or the kind of like liquidy part of the cell outside the mitochondria, and it's going to be transported into the mitochondria by the mitochondrial pyruvate carrier 2. And decarboxylation of pyruvate is going to begin with this multi-enzyme complex, pyruvate dehydrogenase, which we've talked about. And then that, with the help of thiamine pyrophosphate, forming a temporary bond, it's basically going to cause the pyruvate to lose one of its carbons. And then in the second reaction, we're going to have this enzyme dihydrolipol transacetylase. It's going to help this this lipamide molecule attach, and it's basically going to take the TPP from the first step, is going to be removed to form the acetyl group. And then next we're going to have coenzyme A attack through beta oxidation the acetyl group that was donated by the lipamide, while lipamide is reduced and is going to release from the molecule at this point. So earlier in the reaction, the oxidation reaction will occur. We need the cofactor NAD plus to come accept the hydrogen, reducing it to NADH, which will be used later to carry energy in the electron transport chain. And then the molecule we talked about before, thiamine pyrophosphate, forms a temporary bond with pyruvate, which will ultimately give off this carbon dioxide molecule. And in the last steps of the pyruvate oxidation, lipamide transfers electrons to FAD, forming FADH2, and oxidizes lipamide. Ultimately, electrons are going to be donated as well to NAD plus to form NADH, and then the lipamide is reoxidized by dihydrolipol dehydrogenase, so it can basically be reused for further reactions.